Welcome to The Explainer. Today, we're looking at a wild new cancer therapy that works in a totally unexpected way.
So, here's the puzzle. How can a drug that only pauses cancer cells actually make tumors shrink?
Okay, first, a quick biology refresh. Cells have two main jobs. They grow bigger and they make more copies.
In healthy cells, these two things, growth and division, are perfectly in sync. It's a really tight system.
All right, so these new drugs, called CDK4/6 inhibitors, basically hit a giant stop button on cell division.
This specific pause is called G1 arrest. The cell gets frozen right before it can copy its DNA.
But here's the catch. The drug only pauses division. It doesn't tell the cell to stop growing. And this is where cancer's own programming backfires. Mutated genes, or oncogenes, are always screaming, grow. So you've got the brakes slammed on division, but the accelerator for growth is floored. A total mismatch.
And that leads to a fatal condition called toxic cell overgrowth. The cell just gets way too big. This massive overgrowth causes so much internal stress that it damages the cell's DNA, forcing it to shut down for good. So the cancer cell's greatest strength, that unstoppable drive to grow, is turned into its fatal weakness. But how do we know this is what's happening? Well, scientists ran a really clever experiment.
First, they just watched cancer cells do their thing. And yep, they multiplied like crazy into lots of colonies. Next, they added the CDK4/6 inhibitor. And just as you'd expect, the cells stopped making copies.
But then came the twist. They added a second drug, this one designed to stop the cells from growing.
The result? The cancer cells were totally fine. By stopping the growth, they actually rescued them.
And that's the proof. It wasn't the pause that killed them, it was the unchecked toxic growth.
So why does this matter? Well, understanding this opens up a ton of doors for smarter cancer therapies.
And it leaves us with this really powerful question for the future of cancer research.